Hello everybody, I am Dr. Joydeep Banerjee here to deliver the course on advances in agricultural engineering. So, in this course today we will discuss about the topic on biotechnological interventions in agriculture. The course has two different modules what we will discuss today. First one is in module 1 we will discuss about the importance of plant breeding in present scenario, agricultural improvements in different era, pure line theory of plant breeding and backcross breeding method in case of plants. In module 2 we will discuss about the marker assisted plant breeding, molecular markers, restriction fragment length polymorphism and simple sequence repeats in detail and applications of molecular marker in marker assisted selection. So, first I am coming to module 1. So, what are the role of plant breeders in modern day? Nowadays we can see that due to global warming a lot of environmental problems are getting increased day by day, like salt stress, drought stress, flooding. Those abiotic stresses are causing huge loss in agriculture, and in addition to that the global population is increasing at an alarming rate. So, if you see different third world countries, there are lot of poor people are suffering from nutritional insecurity. They do not get enough amount of iron, folic acid in their food. They cannot buy good amount of healthy foods. So, the plant breeders has a huge role to meet up this problem. Certain agencies like ICRISAT, they have developed biofortified pigeon pea. Certain researchers are working to improve the iron content in rice and other crops. In this way, the plant breeders have a huge role to solve this hidden hunger in the society. In addition to that, abiotic stress, every year the soil affected area, the salt affected area are increasing, and if you see this year's scenario in case of India, then more than half of the area was affected by flooding, and a huge amount of area was affected by drought stress. So, this salt stress, drought stress, flooding, it are causing a huge loss in agriculture, and the expected loss is 20 to 50%. So, the plant breeders have certain role to meet up this problem, to combat this problem. To combat this problem, in India a mega rice variety is Sorno. The breeders have introduced a particular gene in this variety to make a new variety named as Sorno sub 1, which is tolerant to submergence. So, in this way the plant breeders are working to combat various stresses. Along with abiotic stresses, biotic stresses are also getting prevalent. Once a plant is affected by abiotic stress, its health condition become poor gradually, and its normal growth and development is hampered. And in this scenario, the biotic stresses like different bacteria, fungus, virus, they infect the plant more easily. The plant breeders have also developed several resistant varieties by the application of either conventional plant breeding or marker assisted selection. Later on we will discuss those things. In certain crops, the breeders can think about development of early variety. Like India is the second largest producer of lentil. It is a pulse crop and it is a winter loving crop. So, if you see the lentil production time, the lentil is grown from November to February. During February it is mostly harvested. Since the last couple of decades, the average temperature in day as well as in night is getting increased in February also, and the farmers, most of the farmers in India, they are facing this problem nowadays. They are seeing that in their lentil fields the pollen abortion is visible, and they are not getting enough seeds due to little bit increase in the temperature. So, if the scientist, if the plant breeders can make a variety which can complete its life cycle little bit earlier with sufficient amount of yield, then that type of problem can be solved. Next, let us see the agricultural improvements over time, how the plant breeders and plant molecular biologists have worked together to improve the agriculture over time. Since long time, the crop plant domestication was done by human beings. Initially, human beings were migratory in nature. They used to migrate from one place to another place across the world in search of food, and thereafter they started crop domestication. And in this picture you can see where different size cob has been shown, maize cob. Here in the extreme left, the earlier sized maize cob was of this size, and thereafter due to course of selection after the domestication of maize plant, they have gathered the large size maize cob. In another picture you can see the wild progenitor of maize, that is teosinte. Earlier this was used by the human beings, and due to selection over time finally we can see the modern day corn. So, thereafter the plant breeders started different working to develop different varieties by conventional breeding methods, and since 1960s, which is the time of the first green revolution, then a lot of semi dwarf rice wheat varieties were came into the picture. Those rice and wheat varieties were highly fertilizer responsive and high yielding in nature. So, finally there was a huge jump in the agricultural production across the world during that era. But thereafter some farmers did not use the fertilizer in adequate manner. They have extensively used the fertilizer, and the soil health condition has been poorer to poorer gradually. If you see the agricultural improvements in recent times, nowadays scientists mostly follow different molecular breeding approaches, either through marker assisted breeding or through transgenic technology. So, in those cases where conventional breeding takes around 12 to 14 years to develop a particular variety, through marker assisted breeding the time period can be minimized. Here by 6 to 8 years we can develop a variety, and whenever a particular gene is not available in the system in a particular species, then we have to go for transgenic technology. Like in case of BT cotton, no cotton varieties, no cotton germplasm was available which is resistant to the pest, infecting pest. So, in that condition the gene from Bacillus thuringiensis was integrated into the cotton genome to make a transgenic cotton to combat that biotic stress. Now, before going to the next part, I would like to discuss about the concepts of gene. So, if we remember the movie Jurassic Park, there we have seen that from the blood samples of a dinosaur we can extract the DNA sequence, and from the DNA sequence gradually we can develop a full fledged dinosaur. Similarly, genes are the functional unit of heredity, means it transfers some characters, some sequences from one generation to next generation, and at molecular level it is nothing but a stretch of nucleotides. In our system 4 types of nucleotides are available: adenine, guanine, cytosine and thymine. So, gene is just a stretches of nucleotide which codes for a functional product. The functional product may be either protein or RNA, and all living beings are composed of genetic material because it is transferred from one generation to next generation. Now, another important topic is allele. What is allele? Alleles are alternate forms of a gene. We all have different genes and they have different alleles. from a particular example it will be more clear. As you can see, let us discuss about the allele of a flower color gene. If we think about allele A, capital A, in capital A, at DNA level few sequences are there, ATG, ATA, CAA, in this way. So, from that particular DNA sequence in our system the RNA will be produced, and thereafter from that RNA gradually protein will be produced. So, the allele capital A may have an alternate allele, allele small a. So, those are alternate forms of a gene, the color producing gene. In the small a allele, in case of TAA, a modification has been occurred in T position, it has been circled over there. So, if a minor change is taken place over there due to random mutation, which is the ultimate source of every variation, then if a change is taken place in the DNA, therefrom in the RNA as well as in the protein, we can see some variation, and once those variations are available, a new allele is evolved. Now, I am coming to the concept of pure line theory. It is a very important breeding method, a conventional breeding method used by several scientists in several crops, and it is possible in case of self pollinated crops. Self pollinated crops, it can produce its own seed, it has hermaphrodite flowers, it has male and female organs. So, what is pure line? Pure line is the progeny of a single homozygous self pollinated plant. Okay. It is the progeny of a single homozygous self pollinated plant. So, I am mentioning here about the homozygosity. So, what is the homozygous condition? In our system we are diploid in nature, means every chromosomes we have in 2 copies. In human 23 pairs of chromosomes are available. Similarly, in different plants each chromosome is present in 2 copies, those are known as homologous chromosome. If a single type of allele is available in 2 homologous chromosome, it is known as homozygous condition. As you can see here that capital A available in 2 different chromosomes, small b is available in 2 homologous chromosome, and capital C is available in 2 homologous chromosome. It means the plant's genotype is homozygous in nature. Okay. So, if a homozygous plant produce its gamete means sperm cell, egg cell, so their single type of chromosome will be migrated, and in that particular gamete its constitution will be capital A, small b and capital C. So, now if that plant is self fertilized, then we can get again the capital A, small b and capital C. So, this is the fact about the pure line theory. So, it is the progeny of a single homozygous self pollinated plant. We have started our experiment with a single plant, capital A, small b, capital C, and the progeny plants are of same genotype. So, it has certain facts like it is applicable to self pollinated crops, all individuals in a pure line are genetically same, variation among them is strictly environmental. As you can see here in the progeny plants, one plant is little bit longer, little bit its height is more. So, if the plant is growing close to some manure source or fertilizer source, it may show little bit enhanced height. So, this is strictly environmental, and genetic variability develops with time. As I mentioned earlier that mutation is the source of every variation, ultimate source. So, if these pure line plants are grown over years after years, sometimes a mutation may cause in any genes and it may cause a change at genetic level. So, a scientist named Johansson, he first provided the pure line theory. He worked with Princess Bean and he started his experiment with commercial seed lot. Okay. In the commercial seed lot, different types of seed size was available. So, first he has grown the progeny row from seeds. He separated those seeds. In line number 1 the largest seed was shown, gradually in line number 19 the smallest seed was shown, and thereafter the plants were grown, and once he took the harvest from each different lines, he found that in line number 1 the average seed weight was 640 milligram. In line number 13 it was of around 450 milligram, the average seed weight of that particular plant, and in line number 19 the average seed weight was 350 milligram. So, thereafter he planned his experiment in two different ways. In first case, from line number 1 he did two different types of selection. In first way he took the largest seed, he has grown it from all the seed producing from that plant, he took the largest one again. In this way he has grown it for a couple of generations. Then from the same plant of 640 milligram of weight, average seed weight, he took the smallest seed, he has grown it, and from the produce he took the smallest seed again, and in this way he has grown it for another couple of generations. And finally he found that when he shown the largest seed, the average seed was around 680 milligram, whereas in case of the smallest seed the average seed was around 690 milligram. 680, 690 and 640, these weights are not statistically too much significant according to that particular study. Then in case of the line number 13, he basically divided the produce in different classes based on seed weight. He divided it on into 200 milligram, 300 milligram, 400 and 500 milligram classes, and thereafter he has grown it for a couple of generations, and thereafter he found the average seed weight in the progeny. It was of around 475, 450, 451 and 458 milligram. Those are also not significantly different from the 450, the starting material. So, based on this study, the conclusions of Johansen's experiment was as such. First, the continuous inbreeding, as it is a self pollinated crop, no pollen grains are coming from outside. So, the inbreeding was going on. So, due to continuous inbreeding, the homozygosity was developed. Next one is the variation within pure line is due to environment, as you can see over here, the largest seed, smallest seed, some variation was available there. In case of line number 13, some seeds weight was around 200 milligram, some was 300 milligram, and this variation was due to environment, not due to genetic constitution. Okay? Because in a pod, once the seeds are developed, then all seed will not get enough space. The seed which is getting developed at the center, it will get maximum space, at the corner it will get lesser space. In this way the seed size may vary, but the genetically they are similar. The third point is the selection within pure line is not effective, as all of them has same genotype. As we have seen here, that in case of line number 13, he has done some selection, 200 milligram, 300 milligram, different classes were made. Similarly, in case of line number 1 also, he made the largest seed and smallest seed selection, but it was not effective. The 680 and 690 was statistically almost similar, 475, 450, 451, those are almost similar. So, the selection within the line, within pure line is not effective. And the fourth point is selection in the original population is effective due to genetic variation available within that. As it was a commercial seed lot, so during a lot of cycle of inbreeding, gradually sometimes mutation have occurred and new variations have been generated. So, now I am coming to a very interesting topic, that is backcross breeding method. So, backcross is a very important plant breeding method in case of conventional plant breeding, and it is the crossing of F1 or first filial generation with one of its parents. And in backcross breeding, we have to cross the F1 with one of its parents again and again, means generation after generation. So, certain requirements are there, later on we will discuss it. First one, the recurrent parent, means which parent is used several times in the backcross breeding is known as recurrent parent, and it is generally a well adapted variety and it should possess all the desirable traits, like it should have all the good agronomic traits except the character for which we are doing backcrossing. From the next slide it will be little bit more clear. The next point is the donor parent should have the specific trait in intense form, means from the donor parent we are transferring a specific gene or a specific trait. So, it should be in an intense form in the donor parent. Third one, the characters should be highly heritable in nature, means once we are doing the backcrossing again and again, the characters should not be highly influenced by the environment. Okay? So, the high heritability should be there. And last one, several generations of backcrossing is needed. Why? I will say later on. So, now we will discuss the backcross method for dominant gene transfer. Now, another important term came, dominant gene. So, as we have discussed about the allele, alleles are different alternative forms of a gene. So, the gene may be either dominant allele or recessive allele, two types of allele may be there. The dominant alleles are those allele which show its character, which show its expression, whereas the recessive allele itself cannot show its character. Okay. So, let us discuss over here about the backcross method for dominant gene transfer. Suppose we have a parent P2 which is over here, it is a high yielding variety, but it is susceptible to a particular disease. Okay. Whether another variety which is P1, it is resistant to that particular disease, but it is a low yielder. So, we do not want the variety which is low yielder, just we would like to transfer the disease resistance trait from the low yielder variety into the high yielding variety. Okay. So, for this reason we can go for backcross, and if the resistant gene is controlled by the dominant allele or capital A, then we can follow this type of procedure. So, let us assume in the non-recurrent parent, means which will be used only once in this backcross method, that is capital A which is parent 1. So, it is crossed with the parent 2, the small a. In F1 or first filial generation, one chromosome will come from here, the capital A, and another chromosome will come from the parent 2, that is small a. In the hybrid, in the F1 we will get capital A and small a. So, as it is a backcross method, thereafter we are crossing the capital A small a with the small a genotype, means the recurrent parent. In next generation, the third year, this is the backcross 1 generation. Here we will get two types of seeds, one will be small a, another one will be capital A small a. So, if it is a disease resistance gene, once we infect all the seeds with the disease, the small a plants will be killed as it is susceptible, only the capital A small a will be survived. So, those capital A small a plant will be again crossed with small a plant, that is the recurrent parent. So, in this way from backcross 1 up to backcross 6 generations we have do crossing with the recurrent parent again and again, and in every year we have to do selection or disease screening to eradicate the small a genotype. So, in backcross 6 generation what we got, the small a and capital A small a, like the backcross 1 generation. Here also we have to do the disease screening, the disease resistant plant will be survived and the disease susceptible plants will be killed. Thereafter, the disease resistance plant will be self pollinated, means if capital A small a is self pollinated, it will make three types of plants, capital A, capital A small a and small a small a. So, then we have to make plant to progeny row. So, in this way Now different progenies are grown, and finally thereafter we have to go to the replicated yield trial with the recurrent parents as one of the check, as recurrent parent have been used several times in the backcross method and it is a good yielding variety. So, it will be used as a reference check, and thereafter in 12th year we have to start the seed multiplication and distribution. So, these are the facts about the backcross breeding methods. It is a very important methods, but it has certain disadvantage, like new varieties will not be very superior compared to the recurrent parent. Why? Because we have selected the recurrent parent which is highly adapted high yielding variety. Just the minor disadvantage of the recurrent parent have been improved through backcrossing. So, the yield will not be too much higher compared to that. So, very superior variety will not be developed through backcross method. Next one, the closely linked detrimental genes may be transferred with the target gene. That is the main problem, because once we will do crossing, then a specific gene will not be transferred. The chromosomal region will be transferred from one plant to another plant. The segment of chromosome will be transferred, and genes are available inside the chromosome. So, along with our desirable gene, a number of other genes can be transferred. So, in this way, along with the desirable genes, some detrimental genes may be transferred. So, sometimes we may get some plants which may be a disease resistant, but poor yielder after backcrossing. And last one is, it is highly time consuming and labor intensive process, because we have to do crossing every year. Every year we have to cross it with the recurrent parent. So, it is a very labor intensive process. So, that is all for today's discussion.